So greetings to all. Thank you very much for your presence. So last week we had the CMIT maize team presenting great insights about the maize research that they are going on and using molecular tools such as the KASP markers, but also the dark tag mid-density panels and other technologies. And today we will have Dr. Eunice Kenzime presenting us her research on common bean. So she will be introduced by Dr. Godfrey Azia. Dr. Godfrey is the Director of Research of the National Agricultural Research Organization, but also the National Crops Research Resources Research Institute. Dr. Godfrey has huge experience in crops such as rice and maize, and in maize particularly applied on urgent matters such as the resistance to Stenocarpella maidis and Fusarium graminearum, resistance to storage wilt, steam borers, but also the MLM disease that is so common now in Africa. So Dr. Godfrey, thank you very much for accepting this request. We are very honored with your presence and time, and I will let you add more into my words and present Eunice to the audience. Thank you very much.
Thank you very much, Anne. I think in the respect of the different time zones, allow me to say greetings to everybody who is online here. And it is my pleasure this evening, this morning, in other places, to introduce our speaker today, Mrs. Eunice Kenzime. Mrs. Eunice works with us here at the Institute based in Namlonge in the legumes program as a bean breeder for about 10 or so years. And she has been involved in breeding beans for various traits, including drought tolerance, micronutrient enrichment, and also pest and disease resistance. So her current work focuses on cooking time, especially the short cooking time, and also canning quality in the bean. And through her research, she has already identified some promising genotypes that can be moved forward to product development. And her work has gone further to develop SNP markers from an earlier study using GWAS to identify those markers, and she has gone ahead to convert them to KASP markers for deployment in marker state selection. And those markers are significantly associated with the traits for short-time cooking and also canning quality. So that is very important. It is with that background, therefore, I have the pleasure to introduce to you Anne Kenzime to give us the presentation this evening on the topic of developing and validation of the KASP markers associated with the short cooking time and canning quality in common beans. Thank you very much. And with that, I hand over to you, Anne, to take it from there.
Thank you, Dr. Godfrey. Eunice, you are the host. Please just share the presentation. Thank you.
Thank you very much, Director, for that introduction. And thank you very much, Anne, for the invitation and for the opportunity to share our work. So I'm sharing my screen.
Yes, we can see
my screen.
Yes, just kindly put in presentation mode.
Is that okay?
Not yet. Yes.
Okay.
Thank you.
Yes. So good evening, good morning, everyone. I've already been introduced by Dr. Asea, so I won't repeat. I'll go straight to the presentation. I'm presenting on development and validation of KASP markers associated with cooking time and canning quality traits in common bean, Phaseolus vulgaris. And my presentation is the outline is going to be background to the constraints being handled, limitations and intervention. I'll talk about development of the KASP markers and their technical validation, biological validation of the KASP markers developed, We'll talk about phenotyping for the two traits, that is cooking time and canning quality traits. Then look at marker, segregation, and Delic effect on the phenotypic mean value for the two traits. Give a summary and then acknowledgement. So common beans are such a nutritionally complete food that no other food comes next to them in providing nutrients such as proteins, carbohydrates, iron, magnesium, and others in such high amounts together like beans. Despite that, nutritional value and benefits, over the years, per capita bean consumption in Uganda and worldwide has been almost constant and raw. This is mainly, in among other factors, due to long cooking time that requires high fuel and water and time for cooking. About 40 to 70% of the world population uses firewood as a source of fuel, which is expensive and also has an impact on the environment due to deforestation and then impact on water that is required for cooking. 1 kilogram of dry beans requires about 7 to 11 kilograms of firewood to cook, and incomplete combustion of firewood is the main source of indoor pollutants, especially to the ladies and young girls that are involved in the cooking, leading to acute lower respiratory infections, and also has an impact on gender, denying them especially time for young girls to look at other activities. So there is therefore need for development of short cooking bean variety or a processed bean variety that will lessen fuel wood needs, impact on consumers and on the environment, reduce mineral loss, and improve the nutritional quality of food. Among the processed bean products, we have cooked beans, pre-cooked, which include dehydrated and frozen, flours, protein constraints, and canned beans. And among those, canned beans are the most widely popular. However, phenotyping or selection for cooking time, both cooking time and canning quality traits, is labor intensive. Cooking time estimation methods, especially by use of the Matson cooker apparatus, which is the most common, has a very low throughput and consumes a lot of water and energy, which makes the process very expensive in terms of resources, time, and requires technical expertise, especially when screening large samples. Similarly, testing for canning quality requires specialized canning facility, requires approximately 250 grams of seed for testing, taking a number of seasons for seed multiplication, or requiring a number of advancements if we are starting from breeding lines. In addition to testing for the two traits, it involves destructive process because during testing you will cook and you don't recover the seed that is used. So this would marker-assisted selection would facilitate evaluation of breeding lines at a seedling stage that would enable selecting only the suitable lines rather than advancing many lines that would require rigorous phenotyping. SNP markers are the most widely used in molecular studies due to their low SC cost, high genomic abundance, biallelic nature, locus specificity, low mutation rates, among others. However, there exists a wide gap between the developed SNP and their subsequent application by breeders. This is because SNP databases provide a large amount of SNP information, which requires professional bioinformatic analysis to quickly obtain useful SNPs for easier way for useful SNPs. So there are a number of matrix ways for obtaining this SNP information, which include a Uniplex, or there are many ways of obtaining this SNP information. And among these, the KASP Uniplex SC system has gained wider popularity because of its ability to combine PCR amplification with fluorescence detection and amenability to high throughput automation. So it is upon this background that we sent 15 nucleotide bases flanking the target SNPs on either side of 17 significant markers for cooking time, 37 for canning quality traits, and 8 for water absorption. The nucleotides for these SNPs were downloaded from the SECAT Africa server for the markers that we identified in our study, but we also included SNP markers significant for cooking time and water absorption that were identified by CGIAR in 2015. So with support from EIB, primers were designed from LGC Biosciences Lab. And for technical validation, we sent one plate that comprised of breeding lines. For the two traits, we made crosses for the two traits involving two parents divergent for cooking time and canning quality traits. Then we included other elite lines for the two traits and sent one plate for SNP verification. Upon this, we obtained a technical report of the 28 markers that had performed well, ranging from good to very good. Then we went ahead to validate these 28 KASP markers in an independent population. And these SNP markers are comprised of 9 for cooking time, 3 for water absorption, 2 for splitting, 2 for appearance, 1 for seed coat color retention, 2 for clamping, 4 for viscosity, 3 for wash drain solids, and 2 for wash drain coefficient. So these were selected based on the verification report that we received. And we assessed them using the two populations I've already mentioned. So we sent leaf discs that were cut from the youngest trifoliate leaves at F2 from the plant populations that were growing naturally in the field at Nakri. And with support from EIB, the bean samples were genotyped with the developed markers at Intertech lab in Sweden. The genotyped plants were advanced to F3, and the F3 seed was planted in RCBD on station at Nakri with 2 rows of 1 meter, and we used 2 replications. F4 seed was evaluated for canning quality traits and cooking time 3 months after harvesting. Cooking time was estimated using an automated Matson cooker apparatus, and canned beans were evaluated using a panel of 8 trained personnel on a scale of 1 to 5, where 5 was excellent and then 1 was very bad. And we also calculated the hydration coefficient from the ratio, which is the ratio of the soaked beans to the weight of the unsoaked beans. Then wash drain weight is the weight of the beans, canned beans after washing and rinsing. And then wash drain coefficient is the ratio between the washed drain weight and the soaked weight of the canned sample. And maybe before I leave that, we evaluated the beans based on appearance. Appearance is the general appearance of the canned beans, and the splitting were interested in getting a bean that does not split after canning, and then the beans that maintain the seed coat color after canning, and then viscosity looks at ensuring that the starch solids do not get dissolved into the canning media, and then beans that do not clump at the bottom of the canning container after canning. So in summary, we obtained significant differences among the segregating populations for cooking time. The cooking time ranged from 40 to 128, and we obtained 28 lines that are cooked in less than 1 hour, 45 that cooked in 1.5 hours, and the rest cooked within 2 hours. Then for canning quality traits, which was evaluated on the scale, which were evaluated on a scale of 1 to 5. I'm looking for a pointer. On a scale of 1 to 5, for splitting, we had around 67 lines that performed above average, because from a scale of 1 to 5, those which scored 2.5 and above, it means they were above average. And for appearance, we got 69 lines that are scored above average, and seed coat color retention 75, clamping 69, and viscosity 67. So marker segregation and marker effects, looking at the clustering of the 28 markers that were developed from the KASP markers, 7 markers for cooking time, and 11 markers for canning quality traits, and water absorption clustered into 3 distinct groups, referring to homozygous genotypes for the favorable allele, homozygous genotypes for the unfavorable allele, and the heterozygotes which had both alleles. The allele frequency for the remaining 10 markers did not have any significant consistent pattern. 7 had either only the favorable or the unfavorable allele. 2 had both the favorable and heterozygote and unfavorable allele. 1 for cooking time lacked the heterozygote, and we had 1 which was uncooled. Generally, there has a high call rate of above 90% for 21 markers. We had 4 which had 80, the call rate of 80%. 2 had a call rate of less than 80%. I've already mentioned that 1 was uncalled. So that table gives a summary of the percentages of how the markers segregated, with the homozygous major allele, the heterozygotes, and the homozygous minor allele for about 15 markers that were segregated in 3 allelic states. And that shows also the clustering of the genotypes on the Y and X axis, showing the red, showing the genotypes that had the hex type allele, and the yellow, the farmer type allele, and then the blue color showing both the heterozygotes. So finally, on the allelic effects, on the mean phenotypic value of the 2 traits that we're testing, that is canning and cooking time, I'm looking for a pointer, but it is not coming. Okay, I've got it. Yeah, this marker, it was significant for both appearance and color, seed coat color retention. And the genotypes which had at least a copy of the favorable allele had higher means compared to the genotypes that had only a copy of the unfavorable allele, and the means were way higher above the average. And the same applies to color retention, where at least the copy that had the genotypes that had a copy of the favorable allele almost scored 4 out of 5, and even those which had the heterozygote. Similarly, the genotypes that had this marker for viscosity, at least the genotypes that had at least a copy of the favorable allele had higher means compared to those which had none. And this marker for splitting also raised the means for splitting compared to those which didn't have the favorable alleles. And for hydration coefficient, we had this marker that was selected from Cincy's study that also increased the hydration coefficient means. And for wash drain weight, it was a reverse. The unfavorable allele, instead, those which had the copy of unfavorable allele had higher weights compared to the ones which didn't have any. So in conclusion, phenotypically, this study generated 28 lines that cooked in less than 1 hour and 35 lines whose scanning quality scores were above 70%, in addition to having high hydration coefficient and wash drain weight. 15 KASP assays developed in this study were effective in discriminating against the different allelic states of genotypes at F2. The genotypes that cooked in less than 1 hour had at least a copy of the favorable allele, and these were 4, but we also had 2 where the unfavorable allele is the one which reduced the cooking time. For canning quality traits, genotypes that had at least a copy of the favorable allele of the markers that I've already shown above, this one for seed coat color retention and general appearance of the canned beans, for viscosity, for splitting, and for hydration coefficient, gave higher means, or these means higher, had an impact on the increment of the means for these canning quality traits, implying that these markers could be strongly linked with the genes controlling these traits and thus having a high potential for application in Mars. Yeah, I want to appreciate EIB for the genotyping support, and CGIAR for giving us the phenotyping platform, Feed the Future, and EBICA for providing the resources for phenotyping. Thank you very much.
Thank you, Emis. Very good presentation. Thank you very much. So now the floor is open for questions. You can unmute yourself directly and ask the question to Eunice. And if there is nobody to make any question, maybe I can start with one, Eunice. So I saw that in your first or second slide, there is a gender component in this work. So how much this is being explored and what is the importance? If you just can give some insights.
Well, so far, because we are still at testing stage, we are involving different gender in the evaluation of these canned beans, especially to ensure that we get their views on what they would want regarding the traits in the canned bean, and also how much they want us to reduce the cooking time of the beans.
Brigitte, you can unmute yourself. Thank you very much, Eunice.
Thank you, Anna. Thank you, Eunice, for the nice presentation. I have two questions about the work. One, how was the validation population related to the verification population, like where the QT were mapped? How close were they? And I have a question about one of the slides where you showed the effects of the alleles. Last slide but one. That one. What is not clear here is because maybe it's a bar chart, you can't see the difference between the different genotypes. How much differentiation was present in the validation data per genotype in terms of the mean and the standard error? Were there any overlaps? How much effect of the favorable allele was represented? Thank you.
I answer this, Anna, first. Okay. Thank you for the questions. I'll start with the second. I will maybe need clarification on the first one. Yeah, for the second question, I only plotted the overall means of the genotypes that had the alleles presented in the graphs. For instance, for the genotypes that had the favorable allele CC for this marker, their mean was 3.6, but I did not present all the genotypes. The majority had the heterozygote. Maybe next time I need to find a way of ensuring that the number of genotypes is captured as low, at the same time their means. This is only showing the mean from the overall score of all the genotypes. Then for the population, I request that you repeat for me the question.
So you said that it was the validation was done in other background other than the population of GWAS was conducted. How were the two populations related?
Oh, okay. For as far as I'm concerned, and I understand when you're validating, you're supposed to use an independent population different from what was used for GWAS to find out whether the markers default still work within this population or are still linked to the trait of interest. But for this study, we generated the population by selecting the genotypes that performed well for cooking time. Then we selected those which performed well and crossed with those which, for instance, cooked for a long time. And the same was for canning quality traits. So we actually selected the parents from the screening from GWAS.
Okay. Thank you.
So Eunice, there is a question from Celestina. Celestina, do you want to ask directly the question to Eunice, please? You can unmute yourself.
Hi, good afternoon. Nice job, Eunice. And I just have just comment. Maybe I was asking which kind of market classes did you use for your parents? I'm not sure. I didn't see well how many population did you use, but I just want to know which bin type did you use? And also, do you know if is there any relationship between the grain color and the cooking time? As maybe black beans cook faster in relation to maybe red beans. Thank you.
Okay, thank you, Celestine. Maybe this I can answer on the GWAS study that we did. For the GWAS, we considered almost all the market classes, including the both small seeded and big seeded. Then we also had the reds, whites, yellow, all of them, because we screened 250 genotypes. And when I looked at the correlation between cooking time and the color, most of the small reds and the small white cooked shorter than the big seeded, either sugar and the red mottled beans.
Celestina,
Thank you.
I don't know if you have any other question or if you want to add something else.
No, thank you. Was fine. Thanks. She answered.
Thank you. Does anybody have any other question or comment? So meantime, I have another question, Eunice. Well, Dragan, okay, before I ask this question, Dragan, please go ahead.
Yeah, thank you, Anna. So, Eunice, I have a comment and a question. So, I would like to acknowledge your work. It's great to see that you are working together with other partners on quality traits, nutrition traits in beans, especially from the perspective of future work and project that we will work together. I think there's going to be more space to collaborate with Anna on genotyping efforts in beans. That's a comment, and I congrats for this work and presentation. And one question, according to your knowledge or someone from your team, when it comes to the releasement of the varieties, of bean varieties in Uganda, NPT trials and requirements, are there any nutrition traits required for releasement of varieties in Uganda, like cooking quality or canning, cooking time or canning quality traits? Are there any requirements for the nutrition traits in Uganda, especially because of importance of beans in Uganda, like around 1 million hectares of growing area? Thank you so much.
Thank you, Dragon, for the compliment. What I can say is that in Uganda, cooking time of beans is becoming a very important trait because the consumers really need bean variety that cooks in a short time. Yeah, previously we used to during our PVS tests, we would carry out cooking tests, but it would not only focus on the cooking time, but would also look at the swelling, the softness, the thickness, other qualities of cooking. But it is this time round that we are emphasizing the cooking time to ensure that it is reduced to at least less than an hour for the varieties that are released. Then also nutrition, we've been working on nutrition, especially iron and zinc, and we have a number of 7 varieties released for iron and zinc. It is just because during PVS there is no way you can analyze for this with the different stakeholders, but we emphasize the once they have an explanation, they usually go for those varieties that have more nutrients. So it is one of the traits that the beans that are released must have in the released varieties that from 2016, I think. We have to ensure that among the other traits, we we have enhanced iron and zinc to start with for the varieties that are released.
Adam, I don't know if you want to add something or if it's satisfactory the answer to you.
No, I would like to thank Eunice one more time. It is okay. Thanks.
Thank you. So my question for you, Eunice, is that what can be more done in this work, and what are the next steps?
Pardon?
So my question is, what can be more done in this work, and what are the next steps?
Okay. I think we have validated these markers in segregating population. One and which had a few samples, actually only 94 samples for cooking time and 94 for canning quality traits. I think we need to evaluate them in a larger population and if possible in advanced lines, yeah, to see if they still work. And also if we can have multi-location trials, yeah, and we ensure we test them in different locations. That would be great, and that's what we are looking forward to.
Thank you. I don't have any other questions. I don't know if the audience have any other question. If not, my advice, Eunice, is that to follow up with Brigitte, Dr. Brigitte. She's in Tanzania or Uganda, if I don't mistake. Brigitte, please correct me. And maybe you can request some advice regarding what she was saying in relation how to present the different alleles. Okay, that is one of the comments. And if there
She
is,
knows me. We are friends.
yeah, yeah. So both are in Uganda, so it is much more easier. And if there is no more other questions or comments, thank you very much, Eunice, for the wonderful work and for the good presentation. Dr. Gottfried, thank you very, very much for your time. I know that you are extremely busy, so we can come to the end of one more seminar of this series. Thank you to all for being here with us. We hope you see you in the next seminar, Focus on Sorghum, and it will be presented by Dr. Andekelil Mawawonj from Tari, Tanzania, with the introduction from Dr. Jurandir Magalhães from Embrapa, Brazil. So Dr. Godfrey, Dr. Eunice, I'm not sure if is this the word, Webale. Thank you very, very much. Thank you to all.
Okay,
You're
very much.
welcome.
Thank you.
Nice evening.
Mm.